Hello, class. We are now looking at the female reproductive system with emphasis on physiology. Previous videos and PowerPoints that you have introduce the anatomy of the female reproductive system. Okay, so where we are, here would be the anatomy that we've already discussed. Endometrium. Uterine fibroids, you can look at these are benign tumors in the uterus that occur in many women, and they can cause bleeding and pain and so forth. This is something you can look at yourself. But I want to get to the actual physiology. So we start with the ovarian cycle. During the embryonic period, germ cells move. Germ standing for germination cells will migrate to the ovaries, just like they did in the male reproductive system. At about 5 months of gestation, which means 5 months of pregnancy, the ovaries contain about 6 to 7 million oogonia. Now, the oogonia basically is the first cell. That's the gonia. That particular cell is 2N, just like you had a spermatogonia. That 2N means it has 46 chromosomes, just like the somatic cells do, the non-sex cells. Okay, then this cell will undergo meiosis, which he talked about, and also I have a PowerPoint on that I want you to look at. That's in ANP1, but also ANP2, right? So each of the oogonia is surrounded by a single layer of flattened cells that forms a hollow sphere called a primordial follicle. Primordial follicle. Now, a follicle, so you'll understand, is within the ovary, and a follicle is an anatomical structure that has some surrounding cells covering the oogonia and later develop through meiosis oocytes. So a follicle is a structure that is not only the germ cell, the reproductive cell itself, but it is being surrounded by other cells. That's a follicle. The primordial follicle is the first follicle in the female. The primordial follicle is the oogonia surrounded by some flat cells like squamous cells. That's a primordial follicle. Of those 67 million oogonia, most oogonia in the female ovaries will die prematurely by apoptosis, which was programmed cell death. So it's programmed to die. The remaining oogonia undergo meiosis, the process I want you to study on. Undergo meiosis, which is this process here, reduction division. So by the end of gestation, that means the end of pregnancy, around the time of parturition, delivery, the oogonia are called primary oocytes that arrest in stage 1. Now the primary oocyte, the name is going to change because the primary oocyte has undergone prophase Roman numeral 1. So it's developing. It's going down the meiosis pathway, and it goes into meiosis 1. There's two meiosis, meiosis 1 and meiosis 2. Well, in each meiosis, review again, meiosis 1, meiosis 2, you have the regular prophase, metaphase, anaphase, telophase, and a cytokinesis. Okay, so it's going to arrest in this prophase 1 and stop there, as we'll talk about. And it stops there because of something called cyclic GMP that we'll get into. Okay, so we go now. Towards the end of gestation, as I mentioned, a female's oogonia begin the meiosis process to produce the primary oocytes. The ovaries of a newborn girl contain 2 million. Now remember, you started with 6 to 7 million, but by the time that you get to the girl is newborn is down to about 2 million. So several have died. By puberty, whenever that occurs, that means basically menarche, when she starts having menstrual periods, is down to about 400,000. But only about 400 of these will be ovulated in her lifetime. Now, where did they come with this number? Let's say a girl is 13 and starts her period and ends her reproductive time in life at around 50. That's around 37 years. So this 400 number is a median value, because if the young lady started at 13 and went to 50, then that's 37 years, and you would multiply that times 12. Now that'll be more than 400, so this is a median value. So in this case, you take in 12 months because the females ovulates once a month under usual circumstances, and divide that into 40, so that's about 33 years. So anyway, that's where they get this, just a rounded number. It's an estimated number. Okay, we go a little further. The follicles, primary oocytes, are contained within the primary follicles that have one layer of cuboidal cells. So I'm going to come here now. What has happened is the primary follicle has a primary oocyte in the center of it, arrested in prophase 1, but the cells have gone from this simple squamous type to cuboidal. By them going to cuboidal, we're going to call this a primary follicle. It's the cuboidal cells. And in the follicle, you have the cuboidal cells surrounding, and you have the primary oocyte arrested in prophase 1 in the center. Now, this occurred because of follicle stimulating hormone. Follicle stimulating comes from the anterior pituitary. And these are cuboidal cells, okay? Now, in response to FSH, the primary follicle, which these cells went to cuboidal, will start to multiply and have a bunch of cells surrounding this primary oocyte. When they multiply, you have several layers of cells, of these cuboidal cells. We term those granulosa cells. Granulosa cells. So now the primary follicle is still a primary follicle, but now it has granulosa cells, and the only difference is that it's a multiple layer of cuboidal cells. Okay? Now, as I mentioned before, the primary oocyte is being arrested because of cyclic GMP, as you can see. These gap junctions coming from the granulosa cells impart to the primary oocyte and disallow it from going any further in meiosis. In other words, it can't even go to metaphase 1. Now, some of these follicles develop vesicles called secondary follicles. When they develop these vesicles, multiple vesicles, still surrounded by a bunch of cells, granulosa cells, then we'll call this a secondary follicle. But the key is you see all of these fluid-filled spaces. Okay. All right, so here's a primary follicle, secondary follicle, a bunch of fluid-filled spaces. Now we go further. Continued growth of this, results in fused vesicles, and these fused vesicles are going to fuse and form a cavity with a bunch of fluid in one cavity, and that cavity is going to be called the antrum. And when the antrum forms, then the name changes to a Graafian follicle. So now, granulosa cells, antrum, Graafian follicle. Now, the cell layers within the Graafian follicles, I'm going to this picture now. This is a Graafian follicle. Okay. Now, these cells here, your granular cuboidal. These cells that are in the ovary that are outside a follicle are called theca cells. These are theca cells. These are the theca interna. The theca interna ones are the ones closest to the granulosa. Then you got the theca externa, which are further away. Okay, but right here is that oocyte, right there in the center, rest in prophase 1. Now these granulosa cells that surround that oocyte itself are called the corona radiata, the accumulation of those, corona radiata. And inside here, let me blow it up a little bit, inside here is a layer of a glycoproteinaceous structure surrounding the egg, primary oocyte, called the zona pellucida. When the woman ovulates, she's going to ovulate the corona radiata and the zona pellucida. Now this attachment here to that from these surrounding granulosa is called the cumulus oophorus, which also is granulosa cells. So all of these that look like that. Continued development, so several of these go and become Graafian follicles, but continued development of one Graafian follicle, this is in a month, let's say, occurs because of stimulation from FSH, estradiol, which is estrogen, and paracrine. Paracrine signals, again, are local signals, local chemicals, paracrine. Okay? And so what will happen now is, we'll go further here. So before we go further, I did a little review. The primordial follicle contains many oogonia surrounded by flattened squamous cells. Okay, that's the primordial we talked about. Some of the oogonia undergo and stop in prophase 1. We talked about that. Okay, primordial follicle. When a primordial enlarges some of the surrounding squamous cells and become more cuboidal, then we'll change the name to primary follicle. So the cuboidal cells making a primary follicle, okay, with the oocyte in prophase 1, okay. When FSH stimulates the surrounding cuboid to perform mitosis, giving more cells, a bunch of cuboidal cells, we call them granulosa cells, okay. When the primary ovarian follicle enlarge more and develop several fluid-filled vesicles, they are termed the secondary follicle. And when some of these secondary follicles grow even more and form the antrum, that's called the Graafian follicle. The Graafian follicle. So that's where we are. Okay. So here's a look at the Graafian follicle. Here's a look at the Graafian follicle. Let me check my time here. Okay. Still have a little more time. Okay. So that's the Graafian follicle there. All right. So now where we go is the secondary oocyte. So luteinizing hormone stimulates receptors on the outer granulosa cells. Okay. On the outer granulosa cells, these cells out here, to decrease the amount of cyclic GMP. Now remember, the cyclic GMP produced by these cells was what held the oocyte in prophase 1. And when you decrease the cyclic GMP, luteinizing hormone did that. That allows this primary oocyte here to go in further into meiosis. And when it goes further into meiosis, we come here. It comes further into meiosis. It's going to go into meiosis 2. It's going to go all the way to meiosis 2, and it will stop at metaphase 2. So we'll go all the way to meiosis 2 once LH stopped the growth, the GMP from being produced, and it will stay in meiosis 2, metaphase 2, and the only way it's going to go all the way to the finality is after ovulation, and fertilization occurs. Let me check my time again. So I'm going to end here.